So I start off with a picture of a world-class weightlifter. I'm trying to execute a lift. Quadriceps of a world-class weightlifter can generate maximal contractile strength of about 1155 pounds. That's a lot of force. And all the force is applied to the patellar ligament right there. That's usually what we think about when we think of muscle, the strength it provides for our body. As an anatomist, when I dissect cadavers and I remove the skin and the superficial fascia, and I can see the muscles, that's usually what I think of. And that's what you should start thinking of. There's a lot of muscles in the body. They've been identified, they have names, and that's kind of one of the struggles in this class, is to learn all these muscles. And most of the muscles you see on the body wall and on the limbs are voluntary muscles. But the great majority of the named muscles, they are most of the quote-unquote gross anatomy. Remember, that means, gross means you can see it with the unaided eye. So that's what you see where you move skin and subcutaneous tissue. And so today, before we start naming the muscles, I want you to know the physiology of muscle tissue. So we have a picture here showing you the physiology, and we have some key terms that introduce the idea of muscle physiology. The first term is excitability, key feature of muscle. Excitability means an excitable tissue means it can be stimulated. Muscles are stimulated by nerves, and the correct way we say that is nerves innervate muscles. That's what we say, make them contract. So what I say on the slide is it's the ability to respond to this stimulation. So in the body, nerves innervate muscles and they're stimulated. And so muscles respond to this stimulus by contracting. Muscles respond to stimuli by contracting. That word means contracting, that word means shorten. So muscles pull, they never push. So when you contract, you shorten. Muscles pull. They never push. So in the previous picture of the weightlifter, the patellar ligament is pulling on the knee joint to extend the knee, okay? In an effort to lift that weight. All right, the next word is contractility. The ability to shorten actively and exert a pull or tension that can be harnessed by connective tissues. Okay, so some things there. I already said that muscles pull, they never push. The key word is tension. If you haven't had physics, you should know that tension is a pulling force. Okay. So I'll paraphrase, muscles generate tension when they pull. So the word tension means a pulling force, and it's usually measured in units of kilograms, depending on how strong the muscle is. It's a very small muscle you're testing in the lab, is probably going to be grams, or kilograms if it's a larger muscle. Grams or kilograms of tension, contractility, they generate tension. So the connective tissues, there's a lot of connective tissues associated with muscles. When you study a whole muscle, we usually mostly just talk about the muscle tissue. But the complete whole muscle has a lot of connective tissues. What I'll mention now is muscles exert their force mostly through the tendons. So muscles connect to the skeleton via tendons. Muscles insert, that's the key word, onto bones via tendons. That's the main connective tissue that does the pulling. Tendons are a dense, regular connective tissue. All right, the third term there is extensibility, the ability to continue to contract over a range of resting lengths. Extensibility. The ability to contract over a range of resting lengths. So the way muscles, I mean, you take your muscles for granted, you're born with them. They're in your body in an optimal position so that when they operate, you can exert maximal tension. So for example, the hinge joint is a good example to illustrate this concept. For example, the hinge joint is, pretend the joint bends at that hinge, right there. And let's say you have a muscle that crosses that joint. Right now it's at a 90-degree angle. You know, here's the belly of the muscle. But there's connective tissues on either end that connect the whole muscle to this bone and this bone. And this is some measurable length. Usually you use length L sub naught, that little knot sign. But anyways, you can measure it to a certain length. And you can contract and lift a load and pretend you're holding a weight. And when you contract, you shorten in a way that moves the load up. Now, you can extend the hinge joint a little bit. Let me draw them back here. Let's say like this. Still a 10-pound weight. But now, the muscle is you've extended the muscle, haven't you? I mean, before you generate force, if you were to measure that resting length, well, let's see if you're awake this morning. Is that a longer or shorter length? It's longer. You can still generate tension. You have the ability to contract over a range of lengths. Okay, and you can extend it all the way and still generate force. There's probably some optimal length where you can generate the most force. Like if you ever had the experience of like doing curls in the gym, and how do you cheat? You kind of like flex a little more. You can generate more tension. So there is an optimal length, is what I'm trying to say. But at any length, you can generate. You can stretch the muscle out, and you can still generate tension. So that's what the term means. Right, elasticity, the ability of a muscle to rebound toward its original length after a contraction. Okay. So this is a good example. You stretch the muscle out. I mean, so you do a rep, and you can kind of like just go back to your original resting length. Just return to that one. Call this step 1. Call this step 2. And the muscle will work just the same. It's not like when you sprain a ligament, it gets stretched out and it's injured. Muscle's not like that. You can still tear your muscles, but this wide range of ability to generate force, you can always go back to your original length, and muscles work just the same. Okay, that's what the term means, elasticity. As long as the muscle's not injured. The muscle can return to its original length. To contract. So the chapter focuses on all three muscle types. We spend the most time talking about skeletal muscle. I spend no time talking about cardiac muscle because we learn about the heart in biology 430, so it's not taught in this class. I will spend less time talking about smooth muscle. But skeletal muscle is the most complex, and most time is given to teaching that in this class. But we can categorize the three different types, so you at least know how the three muscle types differ from each other. So we have skeletal, cardiac, smooth. You can categorize these three muscle types based upon categorized based upon whether the muscle is willfully controlled, right? So basically, voluntary versus involuntary muscle. The skeletal muscle you can control with conscious thought. It's a voluntary muscle. The other two, like your heart, I mean, you don't think to make your heart beat, it just beats, right? So it's involuntary. Smooth muscle are the muscles that line the insides of your blood vessels, your digestive tract, all that is automatic, involuntary. Okay. You can categorize muscle whether the muscle tissue appears striped or unstriped when viewed under a microscope. So the word for striped is striated, or smooth. So smooth muscle is named for its appearance under the microscope. It appears smooth, not striped. The other two are striated. Just like the pictures show. Okay, I mean, it's hard to see with the lights on. There is a band in here for the skeletal and for the cardiac. That's what we want you to see. We'll look at better pictures of these later. The third way you can categorize muscles is where they're located. Whether the muscle is located in the body wall or limbs, or makes up hollow organs, blood vessels. Okay. So the body wall limbs, that's skeletal muscle. Well, obviously cardiac muscle is the muscle of the heart. So many places to find it. Everything else is smooth, like lines, organs, right, blood vessels. And pretty much everywhere else. So we'll focus in this class mostly on the identification of skeletal muscles. First, the physiology, and to understand the physiology, you at least have to know how muscles are organized. So you have a good figure in your book. It tells you how to organize them. So we're gonna, again, this is skeletal muscle organization. So when muscle is cut, like the picture, you can kind of like pull out all the different parts. But if you just consider the whole muscle, I mean, what you see is that the whole muscle is connected to the bone by its tendon. I'm showing you there. Again, the whole muscle is connected to the bone. Inserts onto bone via tendon. If you look at the cross-sectional view and you pull out all the parts, one of the subunit is this level here, it's the fascicle. So what we see is that a whole muscle is composed of many what are called fascicles. Whole muscle composed of many fascicles. So the fascicle is kind of like the next level of organization, the next structural unit. So I guess how I describe fascicle is look what's in it, muscle fiber. So a fascicle is composed of many muscle fibers. So the muscle fiber is the next level of organization. The muscle fiber is the cell level. And it's, well, number 3 here, but it's this whole thing right there. That's a muscle fiber. There's one pulled out of the fascicle. It's a muscle fiber. You can call them myocytes. Myo means muscle. It's the cell level. And we'll spend the majority of the time talking about that today because you have a lab quiz on next time, Friday, and it covers the muscle fiber as well as a bone tissue. So the cell level is important for any tissue. We'll talk about the muscle fiber because they're elongated cells. They're not round cells if you look at the picture, right? They're cylindrically shaped. They have many different properties. For example, you can look at the picture, they're multinucleated. Another feature is they have these contractile proteins, structure number 4, called myofibrils. So I'll say that they contain many myofibrils. That's the main thing in the sarcoplasm, which is the cytoplasm of the muscle cell that you don't see in other cells. These are the contractile proteins. So that's the next level of organization, the myofibril in the muscle fiber. Myofibril. It's the contractile proteins. Contractile proteins, there's 2 main ones we'll talk about, actin and myosin. They're filaments, okay? They're thick and thin filaments, actin and myosin. Actin are known as the thin filaments and myosin are thicker. Those terms are interchangeable. If I say thick filament, it's the same thing as myosin, and thin filament, same thing as actin. So what we see is that there's these little substructures within the length of a myofibril. That's level of organization number 5. It's called the sarcomere. So one myofibril is many sarcomeres in a row. So I'll write that. Many sarcomeres in a row. So sarcomere is actually the smallest level of organization in muscle. describe it as the sarcomere is the smallest structural unit of muscle. The word root sarco means flesh. Mere means part. So you see sarco a lot. Okay, so that's how muscles organize. However, the packaging has connective tissue all around it. So we want you to know the connective tissues that wrap the muscle up. So the whole muscle is wrapped with an epimysium. That's what number 1 is pointing to. The epimysium is pretty much the first thing you see when you dissect. So when you see the muscle, you don't see the muscle, you see the epimysium overlying it. It's a very strong connective tissue. Sometimes it's called the deep fascia, aka deep fascia.
That's that white stuff?
Yeah, the whitish stuff.
That covers your brain as well, right?
There is connective tissue that covers your brain. It's called the dura mater.
Oh, okay.
We call those the meninges. It's also connective tissue, yes. So, deep is a relative term, right? Superficial is the opposite. So if there's a deep fascia, there's also a superficial fascia. The superficial fascia is your subcutaneous tissue, right? It's fatty. But anyways, that's where the term comes from. It covers your whole muscle. The other parts, the fascicle, it's wrapping is the paramysium, and the muscle fiber is wrapped with a connective tissue called the endomysium. Paramysium wraps fascicles, and endomysium wraps muscle fibers. They're important to note. Sometimes kinesiologists call these the series elastic component because they help give muscle that elasticity I mentioned earlier. What you can't see in the picture, what you should know is that these 3 connective tissues at the ends of the muscle, they kind of like all bundle up to form tendons. Okay. Merge at muscle ends to form tendons. There's the point I made earlier. The picture on the left is the cadaver, and that whitish looking tissue that the red arrow is pointing to is a good example of deep fascia or the epimysium, right? This whitish color versus it's dissected away here. Or on this side, there's the rectus abdominis, but here it's covered with fascia. That's the deep fascia. So on our models, some of these are illustrated. Now the models are muscle fibers. So the muscle fibers are wrapped with endomysium. And the endomysium is basically an areolar tissue, and there's some reticular fibers there. So there are the 23 is endomysium. They call it a basement membrane. It's kind of what it acts as. You can call it either one. But these, number 24, is the reticular fiber. Keep the lights off. See a little better. If you look at the file on Canvas called quiz slides, those are the slides I will use for your quiz. When is your quiz? Friday, at the beginning of lab, just like the last lab quiz. Okay. Remember how many were on it in your last one?
24.
It was 12, wasn't it?
Yeah, I was thinking of the slides.
Yeah. And it was fill in the blank, which means you had to spell it, right? Yeah, same thing. Okay. And a lot of the pictures on the quiz slides are these same pictures from the lecture. Okay. So here's the whole model. And so that right there is the endomysium. That's a close-up of it. Okay. On this muscle model, right there, you turn it around, the gray represents endomysium and with a lot of reticular fibers. And we're over there, sitting over there. You'll have time in lab to look at those today. I want to talk about the smallest structural unit, the sarcomere. Moving on. Top picture is 400x. When we work with our scopes, that's the max level I wanted you to use. You can barely start to see the striations at 400x. You can see the multinucleated fashion of a muscle fiber. You can see how this is shaped like a cylinder, not a round cell. When you get to 500x, you start to see more detail of the banding. This is obviously you jump from 500 to 64,000x. You can see the sarcomere. One sarcomere is from this line to this line, and the boundaries are called Z-disc or Z-line. The Z stands for a German word I can't pronounce, so just call it Z-disc or Z-line. Sarcomere. From Z-disc to Z-disc. Pretty much, let me advance the slide here. So what I did was I put brackets as a guide to show you the Z-disc on the 64,000x, Z-disc to Z-disc, that's the sarcomere length, right? But on this one I blew up from the previous slide, there it is there, there it is there. So at 500x, you can start to see how the Z-disc, it kind of bisects this light band. Because let's remember, at 400x, all you see is light bands, dark bands. But you can't really see the Z-disc until you get past 400x. So in striated muscle, you basically have light band, dark bands. And at around 500x, you can begin to see the Z disks, they kind of bisect the light bands. (500x), you can begin to see the Z disks bisect the light bands. Okay. Well, here's kind of an illustration of the book showing you Z disc to Z disc, and it shows you all the contractile proteins that we have to learn. And we see that there's way more than Z disc. There's all these different zones and lines and bands that we have to understand to fully get how the proteins overlap to create the banding, light band, dark band, light band, dark band, like zebra stripes. So just to go down the list, the I band at the very top there called the Sarcomere Structure. I band. Basically, the I band are the light bands in the striped appearance. The I stands for isotropic. You're getting into how the protein affects polarized light. You don't have to understand it because I don't. All you have to understand is the way it affects light is it makes it appear light. Okay. So the light bands are the I bands. You should know that. Well, also know what's contained within the light bands. So the illustration helps us with that. We see that there are these contractile proteins called actin. Those are the thin filaments. There's also these springy ones on the end called titin. So I bands contain actin. Remember, those are our thin filaments, and also titin. Titin they're illustrated like springs. They're on the ends of the myosin filaments, and they kind of anchor them into place on the Z disc. The Z disc is the end. So I'll say for titin, anchor ends of myosin, the thick filaments, to Z discs. So in that way, they're organizing the thick filaments. Titin is. Okay. And titin you see in the light bands. Okay, well then, there's the A band. These are the dark bands. The A stands for anisotropic. Just know that what that means is how it affects light. It makes it appear dark. Okay, the A bands are the dark bands. And the definition of an A band, it's the length of the myosin. So what you see in the A band, it basically spans the width of the myosin filaments. That's the definition of A band. Spans the width of the myosin filaments. So again, the myosin are the thick filaments. So that's the light bands and the dark bands. The other things that you have, for example, the H zone. So the H zone, right here, it pretty much begins where the thin filaments end. And within the H zone, you only have the thick filaments. that's the zone where you only have thick filaments. Notice that the H zone is bisected by the M line. M stands for middle. Again, it's another German word, but let's just say it stands for middle. It's right in the middle of the sarcomere, and it basically bisects the H zone. Right in the middle. Yeah.
Did you mention what bisects meant?
Just cuts it right in half.
Okay.
Yeah. Okay, well, let's see here. Well, we can see it on the real picture there. That's the M line, right there. So what zone is this? H. What is it from here all the way to here? A band. Very good. We'll continue on. We have a few more things. Iban Junction is basically where the A band meets the I band, right there, at that line. Iban Junction. Okay. Right there, where light meets dark. Just note that. At the border of light, dark bands. So in a sarcomere, how many Iban junctions do you got? 2? Yeah, 2 is right. So you got one there, and then one there. They're both within the Z disc, which is the sarcomere length. So you got 2. You got 2 Iban junctions in the sarcomere. So you got 2. Well, this is important for physiology. Usually what we see at Iban junction is a structure called the triad. At each Iban junction, is a triad structure. I'll define what that is. Triad implies how many things, though? 3. So at each of those Iban structures, there's this triple structure. Basically, it's for the release of calcium. Calcium is very important for muscle contraction. So we have to study the triad structure. Calcium, ion, release. Ultimately, for the physiology, you have to saturate this sarcomere structure with calcium so the muscles can contract. Okay, zone of overlap, the next one. The overlap refers to where the actin overlaps the myosin. It's where the banding would be the darkest because you have both of those proteins within it. Actin overlap of myosin. Okay, that's how we define it. It's very important. You have to have that overlap. Otherwise, muscle can't contract. Muscle contracting is an interaction of myosin literally cross-bridging with actin. Okay? And if it can't make those cross bridges, you can't generate force. So you depend on this optimal zone of overlap for maximal cross bridges. So that's what I'm writing. Form cross bridges between actin and myosin. So the last thing I already defined. The length of the sarcomere is between what?
Z to Z.
Z to Z. Very good. Okay, so one thing to note, the sarcomere length changes. So do you see how like top relaxed, it's longer, but when you contract, it shortens. So can you picture in your mind how it's shortening? What you should see is that the filaments are sliding past one another. What we say is the actin slides past the myosin. So there's different ways you can describe from this to that. Well, let's say the whole muscle contracts. What's happening at the smallest level? What's happening at the sarcomere level? What happens to the whole sarcomere length from Z to Z? It shortens, right? So that's one thing I'll say. Sarcomere length shortens. A stationary actin slides past stationary myosin. Actin slides past. Notice that the actin, both actin on either side of the M line are sliding towards the M line. On this side and this side, it's kind of like the actin slides past the myosin, but towards the M line on both sides. I'll put comma towards M line. Now here's the thing. What's happening to the H zone? There's H zone there. Where is it there? It basically shrinks. It's gone. There's no H zone here. Because the H zone is where you only had myosin. But when it's fully contracted, it's gone. So I'll just say H zone shrinks. All right, so on your half sheet, go ahead and talk it out. Identify 1 to 7 sarcomere stuff. So on your half sheet, number it 1 to 7. So number it 1 to 7 and label each one, right? And call it sarcomere. One, two, three, four, five, 6, You get stuck on any of them? Let's reconvene. Number 1. Which think that was? H zone. And what is number 2? The line bisecting the H zone. Right in the middle. M line. Number 3, it's the light band. I band. Number 4, it's the dark band. A. Spans the width of myosin. Number 5, Z to Z. Length of the sarcomere. Number 6, right there where light meets dark.
What was number 3? Can we go on a little bit?
Yeah, let me say 6. I'll go back to 3. 6 is what?
Zone.
Iban Junction. Number 3, what was what again, class? Number 3? Iban. Okay, that was number 3. All right, so we're at 6, and then we had number 7, right there, the darkest part, zone of overlap. Yeah, so those are those 7. That's basic sarcomere structure. I want to move on from that. We have pictures of sarcomere. So these are pictures of the models that I've embedded in the slides, so you can kind of see how the models help us understand these things. Well, I'm pointing to right in the middle of the light band, so I call it Z because that's the Z line, Z disc. This is like right in the middle of the dark band, so that's the H zone. So I'm pointing to the Iban junction right there where light meets dark. And this structure right there is the triad structure that you see at the Iban junction. Okay, there's 3 things. 1, 2, 3. I'll do it again. 1, 2, 3. Okay. These little end sacs are where the calcium is released from. Okay, so let's kind of look at other pictures of the sarcomere. Here's another one. Turn the lights off. You see it a little better. Okay. Well, I have it labeled, and you have to kind of turn your head sideways to see the numbers. But number 1 is a dark, thick filament. That's the myosin. I have it labeled down here, by the way. 2 is a thin filament. That's actin. 3, when they put the 3, they're just going for a light band. So number 4 is the Z line, bisects the light band. Number 5, they put 5, 6, and 7 right here. It's kind of inconspicuous to know what they're going for, but it's supposed to be from here to here, which is A band, number 5. Number 6 pretty much is the H zone, and number 7 is the M line going right down the middle. Okay, so this is straight from the manufacturer's key. What I like about this one is that they kind of chop the different parts of the sarcomere so you can kind of see what's in it there for a cross-sectional view. And so, like for example, pictures in your book also try to teach you this idea. For example, if you cut a sarcomere right within the light part of a light band or I band, you should only expect to see thin filaments only. If you cut right in the H zone, off center, within the H zone, you only see thick filaments. If you cut right down the M line, you can see how the M line actually has all these accessory proteins that are interlinked that help organize the thick filaments. So I'm writing accessory proteins interlink thick filaments. So if you cut in the zone of overlap, you expect to see thick and thin organized together. So be able to, identify these lines and zones based upon the cross section on the models. That's the next slide. So I took pictures so we can kind of figure that out. You have cross section of the H zone. You only see the thick filaments illustrated there. This is the light band, the I band, so that's only the thin filaments. This is right in the M line. You can see the interconnection between the thick filaments at the M line. And this is, again, the zone of overlap, okay? So they all show the same things, the pictures in the book and the models in the classroom. So the match is pretty good. So we're going to step away and take a more of a macro view of the sarcomere and look at it within the whole cell, the muscle fiber. Let me define some terms first. The muscle fiber structure, sarcolemma, is basically the cell membrane of a muscle fiber. Cell membrane or plasma membrane, same thing. Sarco means flesh, okay, so that's why you see sarco a lot. So the sarcoplasm, same thing as the cell's cytoplasm. The triad, like I mentioned before, at the Iban junctions, well, it's three things. It's two end sacs, and that's the easy term. The hard term that it's actually called that I want you to use is the terminal cisternae. Terminal means end. AE, because there's two of them. E pluralizes it. A cistern is like a sac shape. So these are sacs that are filled with calcium. Okay, that's where the calcium is. So that's two things of the triad. The third thing is part of a T-tubule. One, T-tubule. The T stands for traverse or transverse. Because these tubes, they dive deep into the muscle cell to allow the nerve impulse to spread throughout the whole muscle cell. As we'll see. Allow nerve impulse. And allow nerve impulse to spread deep into the muscle fiber. Cool. So I want to define some of those terms before I show you this. Now this isn't even showing you the entire muscle fiber. It's kind of a partial of it. But what you see is the sarcomere. We're looking at a flat 2D picture, but really it's the subunit of the myofibril, which is a round structure. So you kind of see Iban right there, the M line. You see the thin filaments and the Z disc at the very end. There's Z to Z. And then you have draped over the myofibril structure it's something called, what's illustrated blue here, it's the sarcoplasmic reticulum draped over the myofibrils. Write that, please. They're kind of like the endoplasmic reticulum in a normal cell. However, in muscle, these are specialized to be calcium stores. Store calcium. Notice how towards the Iban Junction at the end of the sarcoplasmic reticulum, it's kind of expanded. It's kind of dilated. And those are the end sacs of the terminal cisternae. And notice right in the middle of the terminal cisternae is the T-tubule. Okay. So the end sacs, the terminal cisternae, are a part of the sarcoplasmic reticulum. It's just kind of the expanded ends of it. Let's see here. I'll just put that they're draped over myofibrils. You have to saturate the myofibrils with the calcium. That's why they're draped over it. So when calcium is released, it knows where to go. It's right there. It's draped over it. Now, what releases it? The nerve impulse. So that's why the T-tubule is there. The T-tubule is a good transport system for the nerve impulse to get into. It's literally in contact with the end sacs, the terminal cisterna. Calcium release. So let's kind of look at the triad structure on the muscle pictures, the pictures of the models. Okay, well, on this model here, blue thing, identify. T-tubule. T-tubule. See, they don't keep the colors the same for you, unfortunately. Remember the end sac fringe? The terminal cisternae? On this color, it's that odd beige color. So what if I go like this and I say identify all three things? What is that? Triad. But what if I say identify this beige colored thing and that beige colored thing? I'm going for the two terminal cisternae. Okay. And use the terms terminal cisterna. Figure out how to spell that. And for the blue tube, you could just say T. T-tubule is the complete term. Don't just put T. Put tubule with it. Then it means something. Okay, on this model, what if I say on the quiz on Friday, what if I say identify all this? Where are you going to put it? Triad. Triad. Now what if I just point to the two white things? The to terminal cisternae. Yeah. See, now it's white. Oh, okay. What if I point to all the white stuff in the middle? Where are you going to put it? I'm going for sarcoplasmic reticula. Or if I point to the beige stuff in the middle, sarcoplasmic reticulum. What if I point to just the orange thing? T-tubule. T-tubule. See if you're getting this. All on your half sheet. We're on 8, 9, and 10. Eight, 9, 10. Let me ask you a couple things. On number 8, on your half sheet, identify this white stuff right here in the middle. Number 8, identify. Spelling counts. Spelling counts. Once again, number 8. The white stuff in the middle. Number 9. How about, it's white, but at the end right there. Identify that one. The white structure right there at the end. That's number 9 on your half sheet. Number 10, identify this blue structure. Number 10 on the half sheet, identify the blue structure. Alrighty, what'd you put for number 8? Sarcoplasmic reticulum is what I was going for. Number 9, terminal cisternae. That's the term. Okay. Blue thing. T-tubule. T stands for transverse. All right, so that's kind of getting you ready for Friday's quiz and practice there. Here's a picture of the Triad Structure.
Yeah.
And what I like about this picture, it shows the end sac with terminal cisternae filled with the little red balls, which symbolize calcium. What you can see is that the T-tubule is an invagination of the sarcolemma. So the nerve impulse, action potential, whatever you want to call it, is on the surface. It can get deep into the muscle by going down the T-tubule. Yeah. Photos of the book. T-tubule is an invagination of the sarcolemma. The nerve impulse first travels along the surface of the sarcolemma, then deep into the T-tubule. The nerve impulse first travels along the surface of the sarcolemma. Then deep down the T-tubule. And what the illustration is showing you, when the nerve impulse arrives, there are voltage-sensitive calcium release channels that will liberate calcium. Okay? Within the T-tubule, are voltage-sensitive calcium release channels. They're voltage sensitive, so a nerve impulse will trigger that, right? Okay. So what I wrote on the board is basically on this slide here. T-tubules, infolding, I said invagination, same thing. Infolding of the sarcolemma, action potentials travel down them, which trigger release of calcium from the terminal cisternae. So action potential is another way to say nerve impulse. I haven't really taught action potential yet, but just in your mind, just decode it as nerve impulse for now. And sometimes they call it depolarization, same thing. Different way of saying the same thing. Depolarization. So if I use any of those terms, they all mean the same thing. It's the nerve impulse. Okay. All right, here's a picture of the histology, and look at the red arrow. Turn the lights off. It's pointing to TD. Touchdown. No, not touchdown. For us, it means triad, right? Right there. So this is A Iban Junction, right? Here's the light band, right up against dark band, and that's where the structure is. The two terminal cisternae and the T-tubules where you get the calcium release. That's the histology to confirm the whole triad thing being at the A Iban Junction. Here's an outward picture. It's a picture of sarcolemma. So don't confuse endomysium, which is not the cell membrane, right? The sarcolemma is. You can see these little wells, little pits. Those are the T-tubules looking at it from a top view, right? Here's a picture that contrasts the complexity of skeletal muscle from smooth muscle. Skeletal muscle, let me read from my slide, T-tubules, they penetrate the muscle fiber and surround the myofibrils at 2 points, right? The Iban junctions. Smooth muscle, in contrast, has a more rudimentary and shallow invaginations called the caveolae. Okay, so it doesn't go deep in, it just kind of hits in on the surface. So, skeletal muscle. They have the T-tubule system. Smooth muscle. They have the slight indentations called the caveolae. T-tubule is like a deep indentation. Caveolae is just kind of doesn't go as deep. All right, so getting back to the contractile proteins. So we looked at sarcomere, we looked at the whole muscle fiber structure, focusing on the triad. And, but let's just look at the thick and thin filaments by themselves. And the myofibril, start with that one, the thick filament. The thick filament, I think of string cheese. You have all these little heavy heads. You can peel away a bit of string cheese and you got that. Basically that is, well, read this. One myosin thick filament is composed of 300 myosin molecules. That's what you got there. There's 300 of these. One myosin. Consists of about 300 myosin molecules. So let me restate that. Doesn't make sense. One myosin consists of 300. So one myosin thick filament. So one thick filament consists of 300 myosin molecules. So when we say actin and myosin, I put air quotes around myosin because it refers to the thick filament. But the thick filament is actually 300 of these myosin molecules pictured at the bottom. So 300 of these in one thick filament, that's what I'm trying to say. And to analyze the anatomy of a myosin molecule, pay attention to the hinge and the heavy head. The hinge and the heavy head. The hinge, because at that point, it's a hinge. The myosin head can pivot. And if you notice that heavy head, there's a couple of binding sites. Let me see. So there's the hinge. There's the head, the myosin heavy head as they're called. Sometimes the word heavy is used. Maybe they are heavy, I don't know. Never tried lifting one, but they're called heavy head sometimes. Anyways, that heavy head has this binding site. They're usually called active sites, okay? This is where the myosin actually binds the actin. Well, let's see. I want to say right there is where you bind the active site on actin. Okay, so let me clarify. I don't want you to get confused. I'm going to fast forward, then I'm going to jump back. Okay, this is the thin filament. See where it says active site? That's what I mean. So basically, the myosin head fits right in there. So where I'm pointing to, this is where the myosin head binds active site. It's not the active site, it binds it right there. Okay, that's what I wanted to say. There's another one there too. This is where you usually have ATP. Okay, so a lot of times there's an ATPase that can hydrolyze the ATP. So for that binding site, just put ATP. That's usually what we see there. All right, so that's the thick filament. There is one myosin molecule with a heavy head that can pivot at the hinge joint. It has a place where you can bind the active site. It has a place where you can have ATP. Okay. So the thin filament, what it interacts with, is shown here. It kind of reminds me of like two pearl necklaces and kind of intertwine them. It kind of looks like that. So the actin filaments, think of two strings of pearls intertwined. If you take a pearl, they call that the actin subunit. At the bottom, you can see that the actin subunits have the active sites for myosin attachment, right? That was what I alluded to earlier. I'll say have a myosin binding site called active site. The structure is maintained by this rope or cord called the tropomyosin. Notice that the tropomyosin is covering the active site. This is when the muscle is relaxed and is not generating tension. It's before stimulation. You cover the active site. That way you can't form any cross bridges between the actin and the myosin when the muscle is relaxed. So tropomyosin. The cord covers active sites, when the muscle is relaxed. The other thing is a triple structure called troponin. This little yellow thing right there. There's 3 subunits within troponin. One subunit will bind the actin subunit. One will bind calcium, and one will is attached to the tropomyosin. Okay, so this is actually a very important part of the muscle for physiology, because what you got to do is, if muscle's going to become stimulated, you have to uncover the active sites. So what happens is, look at the top frame. Muscle is relaxed. The active sites are covered. No calcium. But let's say you get calcium release from the terminal cisternae. See how they illustrate the calcium binding? When calcium binds the troponin, it moves the tropomyosin in a way that uncovers the active site. As soon as the active site is uncovered, you'll get a cross bridge formation, and you can get muscle contraction. Okay. Calcium binds troponin. That forces the tropomyosin to move, uncovering those myosin binding sites or active sites. Tropomyosin moves, uncovering, I'll call them the active sites. And after that, the myosin head binds the active site, forming a cross bridge. Myosin head binds active site, forming what's called a quote-unquote cross bridge. And one very important thing on the picture to note is the two arrows. This arrow and this arrow. They're trying to show you that, it's a still picture, but that the myosin head has pivoted in a way that pulls the actin by it. So the actin is sliding by a stationary myosin. I stated that before, but I'm not sure that I'm going to write that down. So actin slides past stationary myosin. Now, they call this the sliding filament theory, that the filaments slide past each other, but really it's only the actin that's doing the sliding. Quote unquote sliding filament theory. Now, when you see theory, you think, oh, is it correct? Yeah, we still call it a theory, but it's how it works. It's accepted.
This is all triggered via a nerve impulse?
Yeah,
Okay.
that would precede it. We're going to take a break, but when we come back, I'm going to start the next batch of slides, just start. And for lab today, I have two labs. And I made handouts in case you didn't print it out. One is for the bone. Bone. Really what I grade is on the back, it's like fill in the blank. Notice I put the blanks on the left side, because if you figured out, I'm left-handed. Yeah, right, so when I grade with my left hand, I go, hopefully you don't miss any, right? So I just grade that for the bone. And for the muscle, it's the same thing. Just fill in the blanks. So it's not a wet lab today, it's just these worksheets, but I'll put them out there so you can grab one and look at them before the break. When we come back from break, a little more lecture, then I'll give you time to fill these out and look at our lab models. So come back in 15 minutes.